Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sano Genetics. Sano Genetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to another episode of the Genetics Podcast. I'm really excited to be here today with Dr. Molly He, who's the CEO and co-founder of Element Biosciences. Since launching Element in 2017, Molly has raised over $600 million for the company, and her career prior to this includes leadership at Illumina, Pacific Biosciences, where she was really at the frontier of both sequencing, protein science, genomics. She's an inventor with more than 100 patents, and you can let us know what the exact number is if it's approaching 200, but we'll explore your journey both from the founding of Element Biosciences, but also your perspective on the field. So Molly, I'm really excited to be here with you today. Thanks for joining us.
Well, thank you so much, Patrick, for having me. It's my pleasure.
I want to start actually with your perspective on the field as a whole. So if we think about DNA sequencing, RNA sequencing, proteomics, and the other assays that you think about that are most relevant for precision medicine, both in health and R&D today. I'd love if you could just give an overview from your perspective of the different key pieces of technology and different things that people should have in their minds for this conversation.
Yeah, that really is a big question. I think I can kind of really expand it into like 2 days of a podcast. But to summarize, I'm truly fascinated about where the field is going. I will talk about the field first and then coming back to, my background and Element and why I'm so excited about that. We are currently at the forefront of precision medicine. We've been talking about precision medicine for many, many years, but we really haven't seen that actually taking effect. Precision medicine on paper, on the words, you already know this meant everybody, they have disease, but they have specific treatment to that individual, right? So that's what we call the precision medicine. We're not quite there yet. And why is that? Well, first of all, we don't have a thorough, predictable understanding of a biology for each individual. And that's going to be extremely complex. It's, understanding biology on the base level is probably relatively easier, but make it predictable. How you would respond to these environmental impacts, how you would respond to certain drugs, that's hard piece, right? So that understanding is still lacking. And the second piece is it's really, really expensive to make drugs today, even today, after all these, AI and aided tools and all that, but still it's expensive. Each drug today, from the beginning of target, discovery all the way to FDA approval, will normally cost you $1 to $2 billion for one single drug. So think about, all these people wanted to have individualized recipe, individualized formulation. How much money would that cost? So that's just not realistic, right? So I think these two pieces really prevented precision medicine from becoming a mainstream. But why it is really interesting now? Well, a couple of reasons. When the technology is continuously improving. And that's why I'm so excited about Element's technology, because I think Element's latest technology can really help to build predictive foundation model to understand and predict biology in each individual person, right? And the second is the drug discovery will become a bit faster, a bit lower cost, and we still do not have the right fully integrated tools yet, but I'm excited that actually the entire field is moving towards that. And I can talk a little bit more about Element's technology as well, why we're at the forefront of making precision medicine affordable.
Amazing. So if people are familiar with, I mentioned Illumina, I mentioned Pacific Biosciences, people are also probably familiar with Olink and SomaLogic and some of the proteomics companies. How should they think about Element and where you fit in with the broader ecosystem of names that people are familiar with?
Yeah, so I think this is a really interesting question because people are very familiar with multiomics, that word, right? Multiomics meaning the omics of all these different molecules, genomics and epigenomics and transcriptomics, proteomics, metabolism. So all these things coming together is multiomics. I think people realize the importance of that because, as I mentioned, I always use this as analogy. Biology is like a river. You do not step into the same river twice because it's so dynamic, right? And when you measure it today, measure it tomorrow, things might be very different. There's some foundational things are the same, but a lot of phenotype will be different. So now that is why multiomics is so important and so interesting. But today's multiomic is very segmented, right? There's great tools for genomic sequences. They're great tools for proteomics. They're great tools for metabolomes, for example. But none of these are fully integrated. So essentially, if you want to measure all these different omics, you will have to take different samples, going through different instruments, going through different workflow, and coming out with different data type, and then somehow informatically or using AI these days to piece all these information together and try to find a unified, integrated meaning for that data. And that is challenging, right? Because the more data doesn't mean better. You really have to deal with all these noises and all that in the integration of these data types coming from different samples. So the benefit or true uniqueness of Element's platform is that we are able to measure all these informations from the same single cell, same workflow, same sample, same instrument, and coming out with one unified data type.
Interesting.
So now you're looking at data that no longer has so much noise that you had to artificially piece them together. And that truly paired multiomic measurement from the same sample, I think is going to give us much better understanding of how biology is done because you're not looking at the, so much noises anymore.
Yeah, that's super interesting. And at a high level, how does that work? Like from a chemistry perspective or microfluidics perspective, like what do you need to do differently to get, it's kind of blows my mind that you can get all of those things out of a single cell, but I guess there's a lot of material in a single cell, right? So how do you do it?
Yeah, the data coming out from this measurement is just like amazing. I think that it will push the next frontier or next bottleneck to be data analysis. But going back to your question about how we made it work, I have to go back to the history of Element, because Element, even today, majority of people think we're a sequencing company. Yes, that is true. However, we're more than just a DNA sequencing company. So that is already embedded in when actually we are starting to build a DNA sequencer. It was not an afterthought. So essentially, I don't know, actually, do you know why this company is called Element?
No.
Okay, so
I didn't do enough research.
People ask
clearly.
me that actually.
Yeah.
So I talked a little bit about that, but not extensively. So when we build this instrument or this platform, we already have the future in mind. And we think our first platform needs to be a DNA sequencer because the market is very well educated. People know how to use genetic information. And as a startup company, we don't have a lot of resources to educate the market and teach market how to use a new platform. So we thought our first instrument should be DNA sequencer. However, we want to do it so differently. So every single technical elements of DNA sequencer is rebuilt from ground up. And with two goals in mind. One goal is how do we increase signal to noise ratio for all of these DNA, for all of these technical elements, so that as a whole, when we put the system together, we have such a high signal to noise ratio, we can use it as a budget to improve cost, to improve turnaround time, to improve data quality and all that, right? So one analogy I always make is seeing stars at night. So if your noise is so low, you can see stars using your naked eyes. You no longer need very expensive telescope or whatever to see stars, and you can pick up stars very, very quickly at night with your naked eyes.
Yeah.
So it's the same kind of fundamental philosophy built into our first generation product, Avidi, that is a DNA sequencer. So signal to noise ratio, that's the first one we invented for. The second piece we invented for is future proof. And that means that can we make all of these technical elements fully compatible to read out protein molecules, to read out protein molecules inside single cells? Can we use the same technical elements to capture cells on the surface, even capture biological samples directly without any library prep? I know this may sound like a pipe dream at the beginning, like especially when we started the company 8 years ago, but that is the vision, that is the innovation that we built into revamping all these individual technical elements, right? So what are the elements we revamped? Well, there's a surface chemistry. It's completely orthogonal to what's out in the field. There's a sequencing chemistry. We use a proprietary chemistry called ABC, Avidi-based chemistry that is based on the avidity concept that we borrow from drug industry. And we also have whole new algorithm for data analysis.
Sorry, what is that concept? I'm not familiar with it, the avidity
concept.
Yeah.
Yeah. I'm happy to got into that, but there are a few other pieces that's also new, like optical system, data analysis and all that. Probably don't have a whole lot of time to talk to that. So Avidi-based chemistry, essentially, let me walk through how sequencing is done on our system. Are you familiar with Illumina's SBS chemistry?
A little bit, but okay. A refresher would be very helpful.
Yeah, so there are many different sequencing chemistries out there, but the one dominant one is sequencing by synthesis, right? So where you incorporate one nucleotide at a time, where this nucleotide has a dye label on it, so you can actually call the base by the color. And also it has a reversible terminator, meaning you only incorporate one base at a time, so it doesn't run like wild in many bases, and that cause problems, right? So you have a very well-controlled system, one base at a time. Once you incorporate, you image, call the base, and you remove the blocker, and you start the cycle again. So that's a typical sequencing by synthesis, right?
Yeah.
So what we do, we call it sequencing by avidity chemistry. And essentially, we break the incorporation step from the detection step. So now we have two steps in the sequencing chemistry cycle. The first step is we actually make this proprietary compounds called Avidi. I'll explain to you later what that is. But Avidi basically has a dye label on it and also has a nucleotide attached to that. So first step is you have 4 different colored Avidi. You bind to DNA balls. That's another piece that we do very differently from the traditional Illumina, which I'll explain in a bit. So we have pre-amplified DNA balls. You bind with 4 different colors of Avidi. Upon binding, the binding is so tight, you can actually image, all these 4 color and call the base. And the trick is you actually remove these Avidi's. It's totally reversible. Now your DNA ball is completely intact. So it's unlike SBS chemistry. After you remove the dye label nucleotide, you actually have some scars left on the DNA. We don't have that because it's reversible. So remove that, you expose the native DNA ball, and then you come in with the dark nucleotide that is reversibly terminated. Now the advantage of that is because it's dark, it doesn't have fluorophore, it's easy to incorporate, it's fast to incorporate. And also because it has a reversible terminator, it's a very controlled process as well. So you can remove the reversible terminator and start binding of your Avidites again as a second cycle. Does that make sense to you?
Yeah.
Okay.
So does that allow you to, what does that allow you to do that the sequencing by synthesis doesn't?
Yeah, so I will explain to you, but I wanted to explain the sequencing workflow first, right? First, you load your linear library that you prepared, and you load onto our flow cell, and we will circularize it on the machine. You don't need any off the machine manipulation. So we'll circularize it on the machine, and we amplify using rolling circle amplification. And this rolling circle amplification, because it's not PCR based, it does not exponentially increase your error, right? So especially it's a linear amplification, so your error is not exponentially amplified. So that's one advantage of our amplification chemistry. And then we go into avidity sequencing chemistry, which I explained earlier. It's a two-step approach. Well, first of all, from that two-step, you already know we do not have DNA scars left. So theoretically, you can, sequence it longer,
right,
and faster, right? Then the second piece, you already know. Actually, I haven't explained. I'm explaining to you now that avidite is a very special compound. It's not a dye-labeled nucleotide like SBS. So this compound looks like an octopus, okay? Mm-hmm. So it has an octopus head, which is a polymer base. And this head, we can up-label fluorophores. So it does not have to be one fluorophore, one nucleotide like the traditional SBS. Now you can actually label multiple fluorophores, meaning your signal is going to be significantly boosted, right? You can have many fluorophores. That's the head. Now you have multiple tentacles coming out with the same linker and the same unique DNTPs at the end of the linker. So that gives you multivalency of a binding of this avidite to multiple strands, multiple copies of a DNA in the DNA ball. And that multivalency is extremely important for improving the quality and reducing the cost. Because think about it, when you have the multivalent binding, your KD is drastically reduced. Your binding affinity is much higher. So as a result, we use 100-fold less dye-label reagent when you compare to the regular SBS chemistry. And we pass along that savings to our customers. So that's one benefit. The other benefit of the multivalent binding is that now the polymerase has multiple chances to make a right call because it has so many different tentacles coming out binding to the copies of same DNA, right?
Yeah.
So even if one tentacle binds to the wrong base, you still have many others calling the correct base, and you can still call the correct base. Now, in the traditional SBS, it's 1-to-1. If it binds to the wrong one, you're done. You're wrong, right? So that actually multivalency of this binding also artificially hide the phasing and pre-phasing information, and thus we can improve the data quality.
So at the end of the day, you get to the point you made earlier, you get much better signal to noise ratio, and you're also getting multiple of these measurements through the same cell. I wonder if you could, you published a preprint relatively recently where you applied the technology in, and I think it was a tyrosine kinase inhibitor resistant non-small cell lung cancer. I'd love if you could just kind of anchor this to one of these real world examples. What were you looking for there, and how did you apply the technology in that particular case? So this is essentially a population of people, but of cells where you're not getting response from the both the first and the second line standard therapies in this oncology, right? If I've teed that up correctly.
Yeah, no, thanks for the perfect segue. So I spent tons of time explaining the sequencing chemistry per se and why this is so amazing. Now, pivoting to sequencing inside cells, because all I talked about is a foundation of sequencing, why sequencing by avidity can be used to sequence we call it direct in-sample sequencing without any library prep. Because think about all these biological samples, the cells, the blood and the tissue, for example, because these biological samples come with a very high background autofluorescence. So for regular sequencing chemists to work directly inside these raw biological samples, the signal to noise ratio is not high enough for that to pick it up signal. That's why we think we're market leader now, because our signal to noise ratio is so high and our fluorophore concentration is so low because we don't need so much dyes to read out our signal. We have actually really great signal noise ratio in the biological samples. And that's enabling us to do all these protein detection, RNA detection, cell paint all together inside biological samples. So that is a very important differentiation. I don't think many of people know. Yeah. As a matter of fact, we have already demonstrated we can sequence inside cells with no library prep for over 150 cycles. And even at the last base, we're still having a Q-score above Q30. And that is unbelievable, right? So this is really attributable to our sequencing chemistry. Now, talking about the TKI story, and this is something we're very proud of, because none of us actually had drug discovery experience. We're just very curious scientists, right? So we wanted to kind of identify a use case for this direct in-sample sequencing. One thing that we always wanted to understand is, can we use this platform to understand mechanism action for certain drugs? And we picked out TKI because it's a very well-known system. There's a lot of clinical studies on that, and there's a lot of drugs of that. So we picked two drugs. One is first generation, the other one's a third generation. And the third generation has much better efficacy and a lower resistance. So we wanted to understand why this is the case. What is the mechanism behind that? So fortunately, we actually made panels, like it's MAPK cell growth and cell death panels that contains 350 RNAs, 50 proteins, including phosphorylated proteins, which is very interesting, and also 6 cell paint markers, so we can follow cell morphology change as well. We then treated the cell line, lung cancer cell line, with these two different drugs at different doses, at different time courses, and loaded onto our flow cell. And in this case, it's 12-well flow cell. And we actually have multiple configuration of flow cells, but these multi-well flow cells allow us to look at dynamic responses, for example, different doses and different time courses from same samples at the same time, under 24 hours with no library prep, right? So we treat these cell line with these drugs, different doses, different timeline, put it onto the flow cell, fix the cells, and then we put into the machine, monitor the entire MAPK pathway, cell death and cell growth pathway, all at the same time. And 24 hours later, we got the results back. And the richness, the results are just mind-blowing. So first of all, we look at, cell paint markers with time, how they change. And then we look at RNAs, then we look at proteins and see how they upregulate or downregulate it with time upon the drug treatment. Yeah. What we found out is very exciting, that we can use these markers to classify cells into live state and death state, right? So what we found out is the third generation drug at the end of the treatment, 24 hours, majority of the cells go to death state and very little left still alive, but it's very different for the first generation. So we know this is fitting into the clinical trial results. But when we looked into further the entire pathway, not just looking at the targets of EGFR, we'll look at the entire pathway of cell death and cell growth, then we found something really interesting. What we found is cyclin D1 gets phosphorylated to a very high level upon first generation drug treatment, meaning that drug actually created a breakthrough pathway for cell to escape apoptosis. And that is why you have resistance upon that drug treatment, gefinitab, and you also have less efficacy, right? So I was like, wow, that's really exciting. Now we're looking at all these different entire landscape of the pathway. We find something interesting. Then, the next natural step is to ask, can we find an inhibitor of that cyclin D1 phosphorylation? If we can, then we can do a combination drug with gefinitab, and maybe these two drugs together would give us much better efficacy and less resistance because we blocked that breakthrough pathway.
Right.
And that's what exactly our scientists did, and that's exactly what we found. And we took this results, so exciting results, to TKI expert. We were told, yes, you're absolutely right. This is not hallucination, because there was a paper published 6 months ago using exactly same combination in human clinical trials, and it saw efficacy. So we're very, very excited, even though we did not know this study was already done in clinical trial, but we actually understood the mechanism of action. And to make it, even more exciting, this is 2 weeks of study using 4 flow cells and 1 scientist with no drug discovery background. So that, I think, can have the potential to totally change how drug discovery is done.
Yeah, that's really interesting. And I guess linking this back to running this all in the same cell allows you to, in a way, correlate all of these markers together in a way that bulk profiling or even single cell on different platforms would be hard to do, right? So you're looking for some kind of pattern of RNA and DNA, DNA not so much, because it's not changing unless you're doing somatic sequencing, looking for some somatic sequencing change, but certainly RNA and protein change. You can look at correlations between those within like subtle cell types, for example, versus looking at some kind of broad pattern across the tissue as a whole. Have I got that right?
Yeah, absolutely. I can give you one use case, which is getting a lot of excitement in the field. We will be shipping in August. We actually doing DIS, acronym for direct in-sample sequencing for optical pool screening. So that's basically perturb-seq, perturb-view, and all that. So the idea is, we are not only able to look at the phenotypes caused by CRISPR perturbation, like, for example, RNA expression, proteins, and cell morphology, how they change, but also we can sequence the guide RNA directly inside the same single cell. Right. So now you have the direct genotype-phenotype linkage from one single run, same sample. So this is tremendously powerful, and we can also do up to 2 million cells per run, and it's very large scale as well.
Yeah, that's interesting. And so how do you think about, like, if you blow up that space of potential applications? It sounds like the really interesting opportunity you see is around drug discovery. I'd love to just hear a little bit more about, like, what do you need, like, because what I'm for the CRISPR case, this is this is like an in vitro assay for the oncology tyrosine kinase. This is like, you have a nice feedback loop where you have, you know what you're putting in one side, you know what you're measuring out on the other side. Are you seeing the most opportunity in these like closed feedback loops where you can really tightly measure the perturbation that you make to the cell and then what the phenotype is? Is there also an application to looking at, I'm thinking about patient samples, like if you were to look at a broad set of people who with immune diseases, and some respond and some don't respond, can you look at their cell populations and try to figure out why? What else can you do here?
Yeah, thank you for the great question. Another thing we're shipping in November or sometime in Q4 is Whole-transcriptome directly inside single cell with no library prep. So now, because there's no manipulation, no amplification, you really preserve the native state of the transcriptome. And at the same time, because we're sequencing inside the transcriptome genes, not just, counting the tags, what have you, we can actually give you a lot more information. For example, we can even do TCR directly inside cells. We can call RNA splicing products. just opens up the whole,
Yeah.
you know, very broad range of application directly inside patient samples, right?
I'm not very familiar with the single cell transcriptomics. Like what are the other technologies that are there today?
Yeah, so the current single cell transcriptome technology is break apart into two steps. One is you have to do single cell manipulation so that you know which transcriptome coming from which cell.
Right.
And then after that, you pull them together and put onto a DNA sequencer, right? So, but you can
You
tell
label
them apart.
them and label them with the cell of origin in some way.
Yes. So
Yeah.
there are many ways to do that. But now you do not have to do that, right?
Yeah.
So basically with our whole transcriptome direct in sample sequencing, you do not need all these manipulations anymore. You can actually look directly inside your biological sample. But I will say it's not a replacement to these traditional methodologies, because those traditional methodologies has their own advantages, because probably, they can scale very rapidly, for example. But I really do see this will significantly simplify the workflow for our customers. And lower the cost as well.
Yeah. Do you imagine a world where this this kind of assay becomes something that is a healthcare system tool, or is it, because I can see two different universes. One is where this is a really powerful discovery tool.
Yeah.
But you figure out like this is the marker that is useful,
Yeah.
and then I'll just measure that in the cheapest possible way in the healthcare system and figure it out. I could also imagine this going the direction where we're seeing sequencing, where whole genome sequencing is slowly but surely becoming the de facto for rare diseases, where you just say we're going to sequence once and make sense of it. I could see something similar in the future where you say actually a single cell multiomic profile becomes a very useful jumping off point for all kinds of things. Like you could do, because you have the genetics, but then you also have lots of these other more like, more immediate readouts of what's the state of that cell today and whatever the disease of interest is.
Yeah, so I first maybe take one step back, explaining to you our platform, which is called a VD24, right? So that's the spatial multiomic platform. But one thing I want to emphasize is this is also a DNA sequencer. So you can do traditional DNA sequencing on the VD24 platform as well, and at a very low cost on your desktop. So I think the list price is like $3.50 per G on your desktop with a very low CapEx as well.
Yeah.
So for that part, as you mentioned earlier, we can already do a lot of clinical research, right, on that traditional sequencing piece. And I think the field is very well educated to that. I don't really need to go through that. Now, the second function of the same platform is what we just discussed. That is the single cell spatial multiomics capability. And for that, you have a great point that I do think this is more of a discovery tool today. And for that to become a clinically relevant tool, it will require some time and market development. Now, the discovery itself, though, I think can have a huge impact to how drug discovery is done or drug development is done. A VD24, with that single cell spatial capability, we believe it can help every single stage of drug development process. From target discovery, for example, we do the perturb-seq type of experiments in the much lower cost, right? That's optical pool screening. And for target validation, we do similar type of assays. And we can understand drug mechanism action, because we have all these panels, and also fully customizable. Everybody have their own proteins and own favorite RNAs. Those are fully easily customizable. So we can understand mechanism action as well. And we can do toxicity studies as well, because we can pick up all these different side effects, right? Because we monitor the entire transcriptome and proteins and cell morphology all at the same time.
Yeah, interesting.
Yeah, so basically we can provide significant value to the entire process. And the goal is, you already know this, 90% of drugs fail in clinical studies because of likely lack of, correlation between preclinical assays and clinical studies, right? Because my hypothesis that one huge contribution for that lack of correlation is because the preclinical study does not look at the entire biology as a whole. So you're missing pieces. You're not looking at the whole picture. Now with a VD24, you can afford to look at the whole picture with much lower cost and much fast turnaround time. Perhaps your correlation, hopefully, will be much better for clinical trials.
Yeah, and I mean, I'm thinking about, I think that cuts multiple ways in both the safety sense that there are things going on in cells that you don't want going on, and you want to figure that out as early as possible, but also mechanistically. And it's remarkable how even the diseases that we think we understand pretty well, every few years there's some landmark paper. I'm thinking about the Huntington's somatic repeat expansion and the understanding of what's happening in different individual cell types within the brain in Huntington's, that I got to imagine that as we get more tools to understand what's really going on in different niche cell types, it's going to change the way that we think about how we go after some of these diseases therapeutically, right? It's sometimes these things are happening in very specific cell types rather than the brain as a whole, as we maybe think about it in a simplistic terms.
Yeah, totally. So one of the things, we talked at the beginning, like how expensive precision medicine is. Now you can imagine how this AVD24 can help, because you could actually do a patient cell line, right? Or even tissue samples into the AVD24, and you can dose it with different dosage of drugs. Because each individual respond to drug differently, and some require higher dose than the others, and some require different combination than the others. So you can actually do this surrogate assay on the VD24 rapidly, right? You can actually dose it differently, in a 48 flow cell. You can do a time course into dose response, and you can do different combination drugs on a VD24 flow cells, and you can monitor the entire transcriptome and proteins of your interest and cell morphology all at the same time. So that I think have the potential to really revolutionize the field.
Yeah, it's super interesting. We talked about Illumina a little bit earlier. I was really curious to ask about, there was recent news that they alleged patent infringement, and I know you all have made a public statement about this, and you're not really able to say lots, I'm sure, about what's going on behind the scenes. But for people who aren't as familiar with the sequencing industry, this is something that happens from time to time. Different technology companies, come after one another for patent infringement around different parts of the technology space. And you've spent time at Illumina, you spent time at PacBio, and I know those companies have probably locked horns in the past. I would just love to have your perspective on that to the extent that you can talk about it. But also, I would just love to hear about how you think about this in the context of building a company more broadly, and the different challenges that get thrown at you by competitors and partners and that sort of thing. Yeah,
no, I was hoping you won't bring this up. I'm just kidding, but that's a good question. I get asked a lot. I obviously cannot talk too much details into this lawsuit, but I will say ever since the news broke out, we got a lot of support from our customers, and most of people are saying, like, congratulations, you're now a legit competitor.
You're in the big leagues now.
So we take that as a compliment, I suppose. But at the same time, we are very confident of our patent position and our inventiveness. We don't have a very large budget for R&D, right? Our R&D budget is probably fraction of the R&D budgets of our larger competitor, but we're very, very proud because our rapid innovation really in the past 8 years has resulted in such a great impact to the field. We're constantly innovating, and this is something we're very proud of. We'll continue to lean on.
What have you learned more broadly about company building? You've been at this for what are we? We're in 2025 now, so plus or minus 8 years roughly, probably plus or minus a few months. If you founded the company in 2017, what would have been some of the highs? What have been some of the lows? And what have you learned?
The learning is tremendous. I can talk about it, days, but I think the highs and lows, I always feel like, because I think I wear this hat, it's called CPO, Chief Problem Officer. Uh, I always feel like, we are constantly in headwinds because it's really, really difficult to build a successful entity in this field that is dominated by one monopoly. And also, when you start to invent outside of just the sequencing field to create your own blue ocean, you're met with different sets of challenges, right? So it's a constant grind. The learning is, I believe, if you ask our exec team, they may give you different learnings, but I really do think people are the most critical factor in your success. Because with the right people, money will come. With the right people, you'll continue to innovate. With the right culture, you will keep the right people. So I think this is the piece. I think it's easily said than done, but I do spend a lot of time making sure, our team is an A+ team, making sure we're all motivated and aligned towards the common goal. And it doesn't mean that we don't argue. We actually argue a lot. We debate a lot. It sometimes gets really heated, but I think this kind of trust is exactly what's behind our success.
How many people are you as a company right now?
Roughly 400.
And I guess you're not all in the same location anymore, right? You're probably spread out. How do you keep that cohesive and innovative culture as you've spread around the world? I think you have people in the UK, you're in San Diego, I understand, but I'd love to hear a little bit about how you managed to do that.
Yeah, so culture is very, very important for the company. And our headquarters in San Diego, roughly, I think maybe a little under 300 people on site.
Yeah.
So that is the piece, we are very proud. We manufacture pretty much everything in-house. So we're very fortunate to have the R&D and operations all together in the same building. The innovation cycle, the learning cycle is extremely fast. So the culture in our headquarter building, I think it's relatively more straightforward to maintain. We have programs, to promote culture, like values award every quarter. And we also, do performance review based on how we follow our values. Yeah. It's kind of embedded in daily lives. But I think, the making sure the field people and all these also represent our culture and the brand will require a lot more work, right? So we do a lot of, weekly commercial calls, and Danae's been doing a great job there. And we also do global sales meetings and what have you. But I think we move really fast. Fast communication, real-time communication is really important.
Last question for you as we wrap up here. Is there a technology that you're really excited about outside of the direct sequencing or RNA proteomics space? I'd love to hear if there's something. It could be something nobody's ever heard of that may never pan out, or it could be something that's just not on everybody's radar yet.
That's a great question. There are so many, but what is the one I'm most excited about?
I particularly imagine that you probably have a lot of contact with early therapeutics, RNA, gene editing, could be. Yeah. Everybody's talking about AI right now. I would imagine you're being used in a lot of closed loop systems where people are looking at, those inputs and outputs, and how do we learn from that big data, throwing some ideas there.
So to be honest, when you first popped the question, AI immediately popped in my head, but I was hesitating because it really feels like a bit too cliche right now.
Yeah.
Everybody chasing AI, but not everybody's AI is born the same.
Yeah.
And I think, AI is just algorithm. Okay. I think the most important thing is not about the algorithm. I think most important thing is the data that feed into your model.
Yeah.
And how you continue to refine the model using this data. That's why I was kind of hesitating. I do think data generation, it's not about quantity. And I'm not a huge believer of just pure quantity that can give you the most precise model. I do think the quality matters.
Yeah.
And the cleanness, the uniqueness of your data points plays a huge role in your success of a foundation model, right? So I think that is a very exciting piece, but I just feel like it's getting too hyped at this point, and not everybody truly understand what it means to have a great AI-driven foundation model. And that's exactly why I'm excited about a VD24, because this is where, I think we can make huge impact here.
Yeah, I think that's a great note to end on. And I see that opportunity in our field very similarly, which is if you look at large language models, you have the whole internet's worth of text, and those are starting to get data constrained now, but they've had a huge run of not being data constrained. I think we have the opposite problem in life sciences, which is most of the key workflows, you get data constrained very quickly. And so, but I do think we can solve that problem by thinking about where are the feedback. There's the broader feedback loop of how do we develop a new therapeutic that gets to a patient and makes them better. But we know that today takes 10 years and $1 billion, like you said. But there are smaller feedback loops with that, which is, you spoke about one earlier, the guide RNA and the morphology of the cell and all these miniature feedback loops. I think there are some of those that we can start to go around very quickly and probably have surprising results.
I think the holy grail for this field, if I have a dream, it is really to mathematically predict biology. I don't know if ever this is going to become the reality, because biology is so complex, right? You're dealing with millions of variables. I don't know if it's at all possible to predict it, but I think to your point, if you confine it in a small scale, a small environment, you might be able to mathematically predict it. So this is something we're truly excited about.
Yeah. Amazing. Well, thank you. This was super educational to me. I learned a lot about new chemistry. I learned about your program. We got to talk about a lot of actually, I think, really interesting applications of this. So thank you for taking the time to be with us today.
Yeah, thank you so much, Patrick. Thank you for great questions.
As always, everyone, thanks for listening, and we'll see you next time. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Sano Genetics on Twitter, LinkedIn, or visit sanogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.